We are pleased to present a video abstract of our review article, Liquid Biopsy Approaches to Capture Tumor Evolution and Clinical Outcomes During Cancer Immunotherapy. This is part of the special review series on liquid biopsies in the Journal for Immunotherapy of Cancer. Why does integration of liquid biopsies represent a critical step towards precision immuno-oncology? The landscape of immuno-oncology is expanding rapidly, initially transforming the treatment paradigm for patients with metastatic cancer, and now both in the adjuvant and neoadjuvant setting. However, optimal patient selection for immunotherapy represents a critical challenge that is intensified by the heterogeneity of clinical responses and inefficiency of imaging to fully and timely capture changes in tumor burden and therapeutic response. Snapshot biomarkers currently used to guide therapeutic selection, such as PD-L1 expression and tumor mutation burden, these rely on analysis of tumor tissue obtained through invasive single-region biopsies, which may not sufficiently capture the clonal complexity of tumors, and also may be problematic in the event of low tumor purity sampling, and importantly, these do not capture the evolving tumor under the selective pressure of immunotherapy. These challenges highlight the urgent unmet need to develop molecularly informed strategies to improve patient selection, enable early and accurate response assessment, and facilitate long-term response monitoring during immunotherapy. To this end, liquid biopsies are emerging as potentially powerful, minimally invasive approaches to monitor tumor burden as cancer cells go through immunotherapy-induced evolutionary bottlenecks and rapidly and precisely capture therapeutic response, a concept that is now prospectively tested in clinical trials. Cell-free DNA, or cfDNA, is released predominantly from cells through apoptosis and necrosis, and a small fraction of cfDNA is tumor-derived, referred to as circulating tumor DNA, or ctDNA. ctDNA can be representative of molecular alterations originating from distinct primary and metastatic tumor sites, as well as tumor subclones. Therefore, ctDNA analysis may offer greater accuracy for the assessment of genomic biomarkers, especially when tumor tissue is insufficient for molecular analysis. And over the past 2 decades, a variety of ctDNA-based technologies have been developed for interrogating a broad range of alterations, including sequence mutations, copy number changes, chromosomal rearrangements, methylation patterns, fragmentation lengths, nucleosomal occupation. And these approaches can be tumor-informed or tumor-agnostic and are being tested in a variety of different clinical diagnostic applications, including early detection of cancer, high-risk patient identification after definitive treatment, genotype-based patient selection for targeted therapies, therapeutic response monitoring, and non-invasive tumor profiling at disease progression. Specific to cancer immunotherapy, longitudinal ctDNA assessments can provide insights into tumor burden dynamics and emergence of therapeutic resistance, which may be detected way earlier compared to radiographic responses. Through rapid and precise interpretation of therapeutic response and indirectly of tumor immunoediting, liquid biopsies may be particularly informative in clinically challenging scenarios, such as understanding of radiographically stable disease. Now, in considering the clinical implementation of liquid biopsies, it is very important to select the liquid biopsy approach based on its diagnostic application and intended use to ensure that biologically relevant alteration types are assessed and that the technical performance of the approach is sufficient in order to minimize the potential for technical limitations such as assay sensitivity or biological limitations such as alterations associated with clonal hematopoiesis. How can ctDNA analysis assist with clinical decision-making? Single and/or serial liquid biopsies allow us to pivot. We can employ liquid biopsies and select and triage patients for cancer immunotherapy based on blood tumor mutation burden or microsatellite instability status, as well as evaluate tumor burden kinetics during therapy in both the early stage and metastatic disease setting. In the adjuvant, neoadjuvant setting, ctDNA-driven minimal residual disease detection, or MRD, allows for early on-therapy efficacy assessment in patients with localized disease, as well as identification of patients at high risk for recurrence after curative intent surgery. Post-operative ctDNA MRD can be used to identify patients at high risk for recurrence and to enrich for the subset of patients most likely to benefit from adjuvant immunotherapy. ctDNA MRD-driven clinical decision-making, it is not straightforward, especially in the setting of ctDNA MRD negative testing. And as ctDNA technologies continue to evolve, we need to broaden our understanding of ctDNA MRD negative in the context of a given ctDNA assay, a given cancer lineage and therapeutic setting, and design ctDNA interventional clinical trials based on landmark or longitudinal ctDNA MRD detection. For patients now with metastatic solid tumors receiving cancer immunotherapy, ctDNA dynamics may be used as an early endpoint for therapeutic response. ctDNA persistence or rise in ctDNA levels has been consistently shown to signify molecular progression. Therefore, ctDNA molecular progression can be used as a quick fail approach to determine treatment duration, sequencing and intensification, as well as potentially constitute a triage strategy for drugs in development. On the other end of the spectrum, the definition of ctDNA molecular response, this is less clear and likely dependent on the NGS assay employed. And a number of ctDNA-driven interventional clinical trials are now underway to test these concepts. The evolving field of liquid biopsies has opened a unique window of opportunity to enhance the clinical potential of ctDNA analysis for cancer immunotherapy through the characterization of additional hallmark cell-free DNA features using integrative approaches. In addition to pre-therapy evaluation of blood tumor mutation burden and microsatellite instability status, as well as dynamic monitoring of ctDNA burden via ultrasensitive high-depth next-generation sequencing approaches, the assessment of genome-wide structural and fragmentation cell-free DNA features, epigenetic patterns, nucleosomal footprints, these may provide additional features to inform the optimized use of liquid biopsies for patient selection, risk stratification, and treatment response monitoring for cancer immunotherapy. In tandem, ctDNA features can be combined with other molecular or clinical analytes in the context of multi-parameter liquid biopsy testing that may further enhance their clinical value. The overarching question remains, are we ready to integrate liquid biopsies in clinical immuno-oncology? While we're getting closer, several critical factors need to be addressed, including technology calibration and prospective testing in clinical trial setting. We envision that not too long from today, liquid biopsies will enable us to make the earliest, best clinical decisions with precision for the increasing number of patients receiving cancer immunotherapy.